So the basic rationale for the PROfound trial was to validate early observation that patients with metastatic castration-resistant prostate cancers whose tumors harbor DNA response defects are likely to respond to PARP inhibition. And so that was the concept that was based on a fair amount of preclinical data, biological data, stand-up to cancer data suggesting that a good percentage of patients will harbor these types of gene alterations, and then early data from clinical trials and as it relates to olaparib from the PARP trial. And so that was the basic rationale. Obviously, AR-targeted therapy is one of the standards of care in this disease, and the intent was to evaluate whether the olaparib will be better than second-line AR-targeted treatment in the context of metastatic castration-resistant prostate cancer. So basically, the clinical trial was designed, so it's a phase 3 clinical trial. At the time when we designed the trial, the most well-characterized cohorts were patients with BRCA1, 2, or ATM gene alterations, and so the trial pre-selected for patients who have the genomic alteration that might make their tumors likely to respond to PARP inhibition. There was a panel of 15 genes, not just BRCA1 or 2 ATM. Because the other genes, we were not sure how well they were likely to respond to the PARP inhibition, we created 2 cohorts. The patients were assigned to the cohort based on their genomic profile, and the tissue was centrally reviewed using a Foundation Medicine assay, next-gen sequencing assay. And then there was a random assignment, open label, to 2-to-1 olaparib versus physician's choice of abiraterone plus prednisone or enzalutamide. And then as patients were treated, they were imaged periodically, and if they developed disease progression by scans based on a blinded independent central review of imaging, then they were allowed to cross over if they were on the control arm to provide them the access to the treatment. The primary endpoint was radiographic progression-free survival in cohort A, which is the patients with the BRCA1, 2, and ATM. And essentially there was a doubling of the median progression-free survival with a 66% reduction in the hazard ratio of progression or death. That was highly statistically significant in favor of the olaparib. Similar trends were seen in the overall study population, cohorts A and B. The trends in cohort B were positive also, but they were not as the magnitude was not as significant, not as big as the magnitude we saw in cohort A. And then we have some data on gene by gene analyses that would suggest, in fact, some genes actually tend to predict for response to PARP inhibition. Obviously, all of these are exploratory at this moment. Other endpoints included objective response rate, which also was significantly higher. It's like about 33% compared to 2% in cohort A, this measurable disease response rate. Time to pain progression, in fact, the median has not been achieved in the olaparib arm, whereas it was achieved, it was under a year in the control arm, so, and the trend was very significant with a very significant p-value. And then there was, obviously overall survival is very important, however the data is not yet mature at this moment, but despite the fact that over 80% in both cohorts that were on the control arm crossed over to the olaparib arm, we actually saw very early separation of the survival curves and trends that are looking very promising. I would refer actually the audience to the poster discussion session where yesterday the data was presented. Basically, the odds of having those types of mutations in the high 20%, depending on where the tissue is coming from. If it's the primary, it's in the high 20%, slightly higher, I believe 30 or 31% in the metastatic disease tissue. Well, I think from the perspective of a physician who is practicing in GU oncology, This provides, if in fact the drugs will be approved at the end of the day, this provides an additional option for patients with this disease. Clearly, we know that cycling, giving abiraterone after enzalutamide or enzalutamide after abiraterone, the response rates are not high and the odds of response are extremely low. And so that within this space, this is actually a promising treatment with a clinically meaningful impact. Obviously, patients have other choices, chemotherapy and other things, but this would be something that would be, I think, quite, what is the word I want to use, desired by patients. Because again, you're pre-selecting and not giving a random treatment. You're pre-selecting for the odds of response. And it's an oral therapy that's generally well tolerated. Obviously there are side effects there, but generally well tolerated, low-grade side effects. I would say from my end, this is likely to become the standard of care in patients who have progressed on frontline AR-targeted agents. Correct, absolutely. And I think that the more we are doing genomic testing, the more we are learning that a good number of patients might be eligible for targeted therapy. Having said that, at the end of the day, you'd have to have the proof that that targeted therapy is going to work. So aside from clinical trials and providing access to patients to that, the positive thing about this, which I think is very exciting, is now targeted therapy can become part of standard of care in this disease.